Well, hello, everyone, and welcome to another in our series of conversations with speakers at the 85th Cold Spring Harbor Laboratory Symposium in Quantitative Biology, which this year had the theme of biological timekeeping. My name is John Ingalls. I'm the Executive Director of Cold Spring Harbor Laboratory Press and also the co-founder of the laboratory's preprint servers, bioRxiv and medRxiv. And our guest this time is Carrie Partch, who is a professor in the Department of Chemistry and Biochemistry at the University of California at Santa Cruz. Carrie, thank you very much for agreeing to do this, and welcome to this conversation. Let me start by taking you back to your entry into this field. You majored in biochemistry, and then you went into Aziz Sankar's lab at UNC. So you just dived straight into the field of biological rhythms right at the moment of your research career. So what drew you to that field of inquiry?
Well, I've always been interested in the way that proteins communicate with one another, signal transduction and how messages are carried through cells. And so when I began graduate school, Aziz's lab was nowhere on my radar, my short list of labs to join. But he ended up teaching one of my initial classes in graduate school on molecular biology. And his insight was really stunning. And I remember thinking he was someone I would want to work with. And so I did a rotation and worked in his lab. And for me, as soon as I learned about circadian rhythms in that rotation, I was hooked. The idea that there's a signal transduction pathway that so faithfully recapitulates time, that it's a clock, to me was the mystery, to really settle into for my career. And we were just at the time in the early 2000s when all the major players had been identified from a genetic standpoint, but really how they fit together and worked was still unknown. So it seemed like there was a lot to do in this area of the clock.
So that's a reminder to all PIs that if asked, they should always give courses to graduate students.
Absolutely.
And is that a message that you have followed in your own career? I noticed from your lab website that you have quite a number of undergraduate assistants and also, of course, graduate students. So do you do a lot of teaching?
Not too much. I do teach a graduate course on grant writing. So so that's one way to interface with students and help them communicate their science and earn funding. But I also do teach a quarter in a year-long series in biochemistry. So for the aficionados who are preparing to study the subject, I get to teach a class in the middle on ligand binding and protein recognition, which is exactly what we do in my lab. And I will tell you, it has been a great conduit for sort of picking out those stars who, have an affinity for the science and who really enjoy it. And so it's been a great pathway into our lab for students who are passionate about biochemistry. And just as an aside, I think teaching does help to solidify not only your knowledge of the basics, but how to communicate that to others. And so it's been a real, I think, compliment to the research to have the opportunity to teach a little bit each year.
Right. So having identified this field as your passion, after your PhD, you then took a sidestep, but a cunning one, I'm sure. And really, you trained in biophysical methods, and presumably you had a plan in mind.
Oh, absolutely. Yeah, I was hooked. As soon as I began graduate school, I really knew that I wanted to work to unravel the molecular details of circadian timing. And as a result of my work in Aziz's lab, I was very focused on the mammalian or the vertebrate circadian clock. At the time, there was really nobody doing that work in the US. So Ava Wolf, I think, had begun to do some really good work on circadian proteins, but she really didn't publish her first structures until 2009 on the mammalian clock proteins. And so other than my now colleague, Andy Lee Wong, no one was doing biochemical studies on circadian clock proteins sort of relevant to that in the mammalian system. So I decided I would Also, I had an affinity for NMR spectroscopy, sort of doing MRIs on proteins and being able to follow biochemistry, but at atomic resolution. And there was really only one person who kind of was the convergence of all of technique and topic, and that was Kevin Gardner at UT Southwestern, who had spent a lot of time studying so-called past domains in a variety of species from humans to plants. And because past domains feature so prominently in circadian clock proteins, it felt like the right union of technique and subject to serve as a basis for future studies. And I was really fortunate when halfway through my postdoc, we recruited Joe Takahashi to UC Southwestern, which made sort of a nice lateral move back into the clocks, a possibility.
So is there presumably these clock proteins, is there something special or different about them from a biophysical point of view that attracted your attention? I mean, are they particularly difficult to handle in that way? Is that why nobody else was doing this work?
I would say you've hit the nail right on the head. That's something we didn't know a priori, that they were really tricky to work with. I can say that now, having worked with cyanobacterial clock proteins, which are a dream to work with biochemically. They're robust to mutation. They purify at large quantities. They're a dream to work with. We really struggle and fight for every advance in the mammalian clock system because the proteins are challenging to work with, and often we still only work with fragments of proteins because working with the full-length entities, there's a lot of intrinsic disorder in these mammalian clock proteins, as is common in many regulators of transcription. And working with large disordered proteins can be challenging. So, it's taken us a long time to build the tools and quality control checks that we put these proteins through so that we can work with them in vitro and feel like we're actually learning something about the biological system.
Right. Well, I want to get back to the mammalian system, but that's a great segue. You mentioned the cyanobacterial system that you talked about on the opening night of the symposium. So I think you've really answered the question, but I mean, you chose that because it's more tractable. Is there a sort of cell-free system in cyanobacteria that you can work with, or did I misunderstand that?
No, so actually, we really got pulled into this field, into the cyanobacterial field, by our friend Andy Lee Wong. And so this is where I'll say that, in my experience in other research fields, both in my postdoc and before, the group of researchers that we've managed to collaborate with in our field has been incredibly welcoming, incredibly open to collaboration. I'm not sure if that's because we span from atomic to human physiology, right? That there's so much done and over such a wide range of organisms that collaboration is often a way to achieve, deep insight. But we got an email from Andy. Hey, I think I've just discovered something really important, this fold switch mutation that eventually was published in Science. And he said, if you'd like to work on it, I think it'd be fun to collaborate. And I said, sure, why not? Sounds like a great idea. And from really that day forward, our collaboration with both Andy and Susan Golden's lab has been cemented. And so, we didn't really make a conscious pivot to that. We were sort of welcomed into that area by Andy and Susan. And so it's been fun to collaborate with them over the last, 5 or 6 years.
Yeah. So can you give, for the audience, a sort of brief summary of what the cyanobacterial system has told you so far?
Yeah, absolutely. So this gets on something I actually didn't address in your question about a cell-free extract. So Takao Konda's lab, back in the early 2000s, made the fundamentally amazing discoveries that this particular organism can still keep time, even in the absence of transcription in vivo. So you can halt all transcription and they will still continue rhythms of circadian cycles. He then used that information to set up a completely in vitro oscillator. So you can take the 3 purified components of this cyanobacterial clock system, Chi-A, Chi-B, and Chi-C, and if you mix them in vitro at their physiological concentrations and give them ATP as an energy source, they will continue to tell time. So I think that system of being able to reconstitute an in vitro clock has represented one of the biggest foundations for mechanistic advances in any circadian clock. I think consequently, we know much more about the ins and outs of the molecular basis of timing in that system because of the ability to reconstitute a clock, interrogate specific interactions and things like that. And so we really worked to build upon that foundation to understand how it is that timekeeping arises. Up until Andy discovered this metamorphic protein that Chi-B actually could sort of transform into a fundamentally different shape, it had really held the field back, because even though we had structures of the individual Chi proteins, no one had been able to capture the complexes of them together to understand how they interacted throughout the course of a day to enact this post-translational oscillator. And Andy's discovery that one of them sort of transforms into a new shape, and his ability to lock it into that shape, this active conformation, really opened the doors to a whole bunch of new insight.
So does any organism with a nucleus have a clock?
Does any organism with a nucleus? So I would say the answer there is probably no. There are some organisms where people have looked very hard to identify circadian rhythms, such as Saccharomyces cerevisiae, and have not really found a proper circadian rhythm. So it's not as though it's a given that any organism has it. I do joke often that it's like a fundamental shortcoming of our field that we don't have this powerful model organism to explore circadian rhythms. Right. And consequently, we now know from work from Ak Reddy and John O'Neill that some human cells that don't have nuclei have circadian rhythms, and that even in enucleated red blood cells, there are rhythms of modification that occur on peroxyredoxin proteins that happen independent of transcription and translation. And how that fits into the kind of common circadian timing is still really not understood well. So I think there's a number of very big mysteries still about the pervasiveness of circadian rhythms.
And what the evolutionary advantage might be to having such a mechanism.
Yes, I think in many cases where people have looked, from plants to Drosophila, and even starting to get some really nice studies in mammalian species like mice, there does seem to be an advantage, of course, in cyanobacteria, as I forget, in sort of coordinating the temporal basis of metabolic processes and aligning them with the availability of sunlight, for example, in species that are photosynthetic. So there does seem to be an advantage, although there's beautiful work from Carl Johnson showing that if you don't have a rhythmic environment, then having a clock no longer provides that advantage.
So that was going to be a question. If we look at, say, very deep sea organisms, where one assumes they are divorced from sensing the diurnal rhythm, has anyone looked at creatures from the deep sea to see if they mirror their more surface counterparts?
So there is work done, I think, by a number of folks, not so much maybe deep sea, but cave fish. So I've heard an incredible talk by Nicholas Folks looking at Somalian cave fish that live in this completely, buried lake, subterranean lake, that no longer have eyes, no longer have coloration, they don't have a light-dark cycle. But in some cases, they still have this sort of, they're held on to this grasp of circadian rhythms, because even if it's no longer perhaps aligned to the light-dark cycle, there could be other factors locally that help entrain them, such as the availability of food, as species may migrate up and down within a water column, so they can almost piggyback, right, on the ability of other species to sense light dark through sort of open holes where they can attain food, as well as the ability that even just running a little bit out of sync with this light dark cycle still gives the physiology within your body the opportunity to unite and synchronize, right? So there have been many studies in the mammalian field that disrupting clocks in particular organisms leads to disease state, right? So, I mean, this is very far afield of what we work on, but we know that coordinated physiology is one benefit of circadian rhythms. Right. So you might imagine for these cave fish who no longer see the light-dark cycle, that even if they no longer step in line with the light-dark cycle, that having that coordination provides some benefit.
Right. Well, I think we could talk about these things all day, but we have limited time. But I really would like you to talk about your lab's interest in translational work. You know, I mean, we're all very familiar with these awful disorders where people are so challenged by sleep disorders. And I think your lab has an interest in exploring some of those deficiencies and perhaps therapeutic interventions.
Absolutely. So, the interest of this actually dates back to my very first year in graduate school. And this is when Louis Tachek and Yung-Hae Fu first published the first human allele in a circadian protein that caused a sleep disorder, right? So they published a mutation in the period protein that causes familial advanced sleep phase syndrome, which is akin to being an extreme morning lark. So these folks go to bed maybe at 7 p.m. and wake up at 4, perfectly happy, right? Pretty extreme with respect to most of us. And so the idea that, mutation of a single amino acid would disrupt the timing of the clock from its approximately 24-hour timing in humans down to 20 hours, a 4-hour change in the timing of the clock. As a biochemist, that gets your pulse racing, right? To understand the basis. And so I would say that at the outset, studying the way in which, human alleles affect the clock is providing extreme mechanistic input by allowing us to dive into the key steps that control timing. And once we identify how those steps, feature in the molecular mechanism of the clock, in some cases, they're highlighting strategies to help people, right, who may have this. And so the PER mutation that was first identified is actually a pretty rare mutation. I don't know the absolute frequency, but we'll say it's something like 1 in a million. It's rare, but very informative from a mechanism standpoint. But we were very excited to see work coming out of Mike Young's group in 2017, identifying a prevalent mutation in the clock gene Cry1. And this is prevalent, the prevalence depends on your genetic background. If you're of European descent, it's something like 1 in 75 people. So this is, quite abundant. And, when I would talk about this to my undergraduates, I could look at my classroom of 200 people and say, there are some of you that harbor this and, that have late night, sort of propensity. And this really was a place where the interest dovetailed with my sort of long-running fascination with the cryptochrome proteins. These are the proteins that I began studying in Aziz's lab, back in 2000. And I don't know if I probably haven't mentioned this yet, but one of the reasons that Aziz had gotten interested in cryptochromes is that they were related to an ancient DNA repair protein called photolyse that he had worked on for many years. And finding these related genes in mammals kind of sparked an interest in him to see what they did. And he was one of the first groups, along with Joe Takahashi and Bert Vanderhorst, to show that they were essential for mammalian. Just like the DNA repair enzyme. And then they continued on for a little bit. And that's actually what I tackled first as a graduate student, to show that this extra bit that was specific to cryptochromes, I studied it and found that they were so-called intrinsically disordered. And this is a really common concept now in 2021. But if you go back about 20 years ago, people were just starting to talk about proteins that by default, by definition, lacked structure. So, I had identified that these so-called tails of cryptochromes were sort of the best I can describe as a floppy spaghetti noodle, right? They're really flexible and dynamic. We didn't know what they did or how they contributed to the clock, but that was what we had sort of figured out back in 2005. So now you fast forward to Mike Young's study that alternate splicing induced by a 1 base pair change in your genome deletes part of that disordered tail to make you a night owl. So it just seems like such a great question to tackle with biochemistry, that modulation or changing this tail had such a powerful effect. So my student, Giancarlo Perico, took this project on and was able to show that the piece in the middle that gets deleted, which is encoded by the 11th exon of the gene, actually acts as sort of an autoinhibitory module to tune how tightly Cry can bind to its clock protein partners. So in its absence, Cry binds more tightly it basically keeps the transcription translation feedback loop running for longer, which then translates into a delayed release of melatonin, as Mike found in his lab. And because of that, because identifying that the tail regulates a specific pocket on the protein that we had identified and studied for some years, it now opens the idea that if we can find small molecules that target this pocket, we could help restore, proper circadian timing to folks who just cannot, go to sleep at sort of a typical bedtime. So those studies are ongoing now. The job of academic drug discovery is a complicated one, few resources and big challenges. But we've been pairing with a company to try and identify molecules that could regulate Cry1 to control circadian timing in folks with delayed sleep phase disorder.
Wow, well, that's exciting. You're clearly optimistic that you can find something that might work.
It's a challenge, but it's doable. In my postdoc lab, working on the hypoxia responsive proteins, so working on a different family, we were able to find molecules that regulate the transcription factor, and those are now in phase 2 clinical trials. So it is doable from a perspective to identify molecules in an academic lab and move them, down the process towards translation. Right?
And certainly in my experience of talking with graduate students, they are really excited these days when biology turns out to have such a practical application. So I'm sure you are finding your young people very engaged in that kind of inquiry.
Absolutely. Although from the perspective of a mentor, to have someone jump all into drug discovery as a dissertation project is pretty scary. So I usually try and balance, right, this idea of having a sort of a hypothesis-driven project where we're really chipping away at that mechanism alongside something more exciting but possibly riskier.
Well, as an academic mentor here, it's very reassuring to hear an academic mentor, a scientific mentor like yourself, being aware of the fortunes of the student concerned. As you say, you want a project that something will come out of, as well as the excitement of the unknown. So, Carrie, this has been a great pleasure. Thank you very, very much for doing this, and all the very best in the future. We look forward to new exciting things coming out of your lab.
Thank you so much, John.
Thanks a lot. Bye-bye for now.
Bye-bye.